Welcome to bi-weekly ESC Academic Talk Series. Today we have 3 PhD students presenting their research. Let us begin the session with Twinkle. So she'll be presenting online today. And Twinkle is a PhD candidate and PMRF scholar at IISc, developing optical and electrical systems for myocardial fibrosis characterization. Her research interests include biomedical optics, bioelectronics, and healthcare technologies. Over to you, Twinkle.
Thank you. So good evening, everyone. Thank you for the introduction. So today in this PhD research colloquium, I'll be presenting my research on polarization-based optical system for myocardial tissue characterization, which basically will give you a flavor of what kind of healthcare technologies related development we are doing in our department. And to begin with, I am working with Professor Hardik J. Pandya, who is an associate professor in the department. And this work is also in collaboration with the clinical collaborator, Dr. Prasanna Sima, who is a cardiothoracic surgeon in Sri Jayadeva Institute of Cardiovascular Sciences and Research. So before I begin with the actual pathological condition we are working on, let me give you a brief idea about why there is a need to work on cardiovascular diseases. So if we see the global burden in the entire world, so cardiovascular diseases are named as the world's number one killer in terms of the World Heart Federation statistical report that has been published. And it is projected that the CVT prevalence is expected to rise with 90% from 2025 to 2050, with mortality rising. And if we talk about the other statistics which has been published, it says that the CVD mortality rate is expected to increase by 73.4% by 2050, with projected CVD deaths which were 20.5 million in 2025 to 35.6 million in 2050. So the need of the hour is to develop some sort of healthcare technologies by which these rates can be decreased at a faster pace. So in that direction, what we are doing is we are into developing a healthcare technology which basically targets a cardiovascular pathology, which is not just a disease. It is just something which is a hallmark of major cardiovascular diseases, and that is named as myocardial fibrosis. So it is a formation of a fibrotic connective tissue in response to injury, where the accretion of extracellular matrix proteins start happening, which over the period of time when it starts accumulating, it leads to permanent tissue remodeling. And if this tissue remodeling becomes permanent, it can lead to a permanent mortality in case of the heart, which over the period of time can also lead to heart failure, ventricular dysfunction, valvular diseases, etc. So instead of targeting a disease, we tried targeting a pathological condition, which is a common area for most of the cardiovascular disorders. So the scientific rationale among this was, if we try to characterize this pathological condition, we will be able to characterize the cardiac tissue composition. Then we will have a lot of information about the tissue, and accordingly we can develop a technology which can be used either for surgical interventions or by the cardiothoracic surgeons during the intraoperative settings. Because this degree and the extent of fibrosis that happens in the heart, it is a key indicator for disease development and also for the case of the heart failure. And you see the right side here, it shows a healthy heart where we have a lot of cardiomyocytes, which are the cells, the major cellular composition in the case of heart. But the ECM, the extracellular matrix, is devoid of any collagen or little collagen. But in the case of a hypertrophic heart, you can, in simpler terms, it is a diseased heart. What we see is the cardiomyocytes in some part of the area, they die, and they are majorly aligned with the something called as collagen. And this collagen leads to the stiffening of the heart tissues. And if this stiffening becomes permanent, it will either affect the rhythmic activity of the heart, it will either affect the valvular displacement. For example, like if we see here, if this is a normal valve, it is a normal condition of a valve which is open. But on the other hand, if we see the diseased valve, it will not be open perfectly. There will be some sort of extra stenosis, which is the deposition of this collagen across the valves. So the entire amount of blood which we expect to flow from one chamber of the heart to the other chamber of the heart, it won't flow perfectly. And on the other situation, if we see the closed valves, so it will not be closed properly. Either it will be leaky or it will be showing doing some sort of backflow of the blood. So that was then overall research focus, or the research domain that we started working on. And in this direction we explored myocardial fibrosis. But to start with, we started with trying to see that what are the areas where it is already being assessed, and what kind of technological gaps exist where we can actually dive in to develop a technology. So in that area, what we saw was, in case of the myocardial fibrosis assessment, there are a lot of gold standards and other techniques which are being used. But one of the other technique, they had a lot of limitations, and especially the gold standard which is being used till now, which is called as endomyocardial biopsy. It is a very it is a highly invasive procedure, and it has a lot of sampling errors, and it is something which is a time taking process, because a tissue resection happens, then it goes to the lab, then staining happens, then the collagen assessment happens. So it takes a lot of time for the surgeon to actually decide upon that how much fibrosis has happened in the heart. So to take these disadvantage as a positive thing for us, what we assessed the gap was, a tissue level characterization can be a useful modality, but we require something which in terms of technology can have an intraoperative guidance, which can aid them in the intraoperative settings, wherein these kind of techniques which were already available or which are in use, they are not longer in use. So we wanted to do something wherein we can reduce the risk of incomplete removal or unnecessary tissue damage. So we dived into developing a technology wherein we use some optical based modalities to assess the tissue characterization. And also, when we studied about these clinical and preclinical techniques to assess fibrosis, we found that these are something which cannot be used intraoperatively. And also, if they can be used intraoperatively, they were either costly or they were not reproducible. They were very much dependent. So there was a gap which we wanted to fill. So to start with, we started first exploring the polarization properties of heart tissues, wherein we developed a tool like this, which was based on transmission mode spectroscopy. And we tried in finding out the degree of linear polarization of myocardial tissues, and wanted to investigate that whether this degree of linear polarization can work as an optical biomarker to differentiate between a normal tissue and a fibrotic heart tissue. So for that, we developed this system, and the principle was like, we find it out that how the unpolarized light which was emitted from the tissue, uh, from the LED source, how it can be linearly polarized. And when it comes to a tissue which is normal versus a tissue which is fibrotic, how much light it depolarizes. And accordingly, we tried quantifying the degree of linear polarization. And the science behind this was, lesser the degree of depolarization of light, more will be the depolarization, more will be the scattering. And that actually maps to the tissue heterogeneity, which we were looking for the normal heart tissues and the fibrotic heart tissues. These are the findings that we preliminary did on n is equal to 17 human heart tissues. Seven tissues were taken from the cadaver bodies, which did not die due to heart disorders, and 7 subjects were with related to rheumatic heart valve disease, and 3 related to the myxomatous valve disease. Because we didn't wanted to just do a research on something which is just one disease oriented. We wanted to dive it into multiple domains. So what we found was, the detected voltage and the corresponding degree of linear polarization was very much lesser for the control subjects compared to that of the disease samples, which related to the signs which we were looking for to differentiate between normal and the fibrotic tissues. And also we tried seeing that how these kind of degree of linear polarization can be related to the tissue thickness. So we tried experimenting with different tissue thicknesses from 0.6 mm to 2.6 mm, for control tissues, for RHD samples, and for MVD samples. And what we observed was, as we were increasing the tissue thickness, the DLP of the control subjects was reducing with a more slope compared to the DLP with the subjects who had RHD and MV. So slope could be used as a, which were observed to be distinct, indicating the delineation between the tissue samples. So this was the preliminary study that we did in order to just look over that whether polarization properties of light can be taken forward as an optical biomarker to distinguish between normal and fibrotic heart tissues. And later we actually, extended this study to further explore this to a greater extent. But all the results that we got from this study was presented in a conference, which is Optical Sensors and Sensing Congress. And also the preliminary results were published in Journal of Biomedical Optics. Now to dive on to further, what we did was, because the system that we tested before was something which was transmission based, but our main idea was to come up with something which is an can be used intraoperatively, or which can be used by the cardiothoracic surgeons to actually test on the heart or the heart tissues. So a reflection mode configuration based system was a more better choice than the system which we developed earlier. So what we did is, in order to explore more about the polarization properties of myocardial tissues, we developed an Mueller matrix-based optical system, which was integrated with an optical fiber-based reflectance probe, which has two transmission systems, two polarization systems. One was meant for transmitting the polarized light. One was meant for receiving the polarized light, and everything had a set of linear polarizers, light emitters, detectors. We had collimators and an optical fiber-based reflectance probe. Now, this reflectance probe was something which has polarization maintaining optical fibers, so that if we are incidenting light on the heart tissue, the polarization is maintained, and whenever it is reflected back, the photodetector is able to catch it forward. So this is the schematic of the optical system that we developed. And these are the rendered images of how the actual optical system looked like. This shows the exploded view with the different transmission polarization system, the PCB which was made for the laser diode and was made for the photodetector, along with the stepper motors which were meant to rotate the linear polarizer at different angles. Why that was done, I'll show you in the mathematical model, which is here. So as I told you, it was an optical system which was based on Mueller matrix polarimetry. So the idea was not just to shine the light at one angle on the tissue, but to shine the line at multiple angles. And that was done using the stepper motors that I showed before. So this is how the Mueller matrix looked like. Basically, every element of Mueller matrix mapped to a combination of optical intensities. So this HHHV basically represents the state of first linear polarizer and the state of second linear polarizer. And using those optical intensities, we computed these Mueller matrix for all the tissues that we tested, followed by quantification of these optical parameters, which actually relates to the different polarization properties of the heart tissues. So we tested for anisotropy degrees, depolarization, polarizance, diattenuation, and the angles, because in angle of maximum transmittance of polarizance and diattenuation. Why? Because in case of the heart tissues, we have collagen, which are like the aligned fibers. So they also have some horizontal axis, some vertical axis. So we also wanted to explore those optical properties of the heart tissues. And this is how the experimental setup looked like. So the optical system with an enclosure that was meant to keep the reflectance probe. And this inset basically showed that how the heart tissue was kept, and how it was being touched with the tip of the reflectance probe. These two pictures actually shows the left ventricle heart tissues and the right ventricle heart tissues, which are excised from the cadaver bodies and also taken from the patients. So the type of tissues that we tested were 56 human heart tissues, out of which 20 healthy heart tissues were tested. Healthy heart tissues were all excised from cadaver bodies, and 36 fibrotic tissues were something which were excised from the patients which had some sort of fibrotic disorders. So that was something which used to be given by our clinical collaborator. And this is how the experimental procedure looked like. So for example, while transporting it from the hospital, the heart tissues were always carried in a saline solution. Then they were taken out through a tweezer, letting the tissues to equilibrate, and then proceeded with the optical measurements, putting in the enclosure and doing all the readout. Now, after we got the all the quantifying parameters for the healthy and the fibrotic heart tissues, we did a comparative analysis. So the first study that we did was just the comparison of healthy and fibrotic without seeing that it belonged to which ventricle. So we got these interesting findings, which I will be summarizing in the last slide. And then we tried exploring just the LV tissues, that how the left ventricle tissues behaved for healthy versus fibrotic. And then we did a study on the RV tissues, the right ventricle tissues. So when we did the statistical analysis and all the research findings, what we observed was when we were treating the heart healthy tissues versus fibrotic tissues, most of the optical parameters were significant. And in case of healthy left ventricle tissues also, we observed significant optical parameters, which were very similar to what we got for when we did the without ventricle study. But for the case of RV tissues, what we found was the optical parameters were not at all significant. So we got back onto exploring that why RV was not behaving the way we wanted it to be. So what we found out that when we looked onto the clinical impression of the patients from which we have taken the fibrotic tissues, what we found were they were the ones which had rheumatic heart valve disease or myxomatous heart valve disease. So this is something which majorly affect the left ventricle and not the right ventricle. So we expected that the left ventricles are fibrotic and not the right ventricle tissues are fibrotic. So that correlated our study results with the clinical significance or the clinical impression of the tissues that we were testing. And also other interesting findings which we got was the fibrosis across both the ventricles was associated with increased anisotropy and polarization dependent responses. So this is very much consistent with when we actually study the science behind the heart tissues, that we expect that in case of fibrotic heart tissues, there will be accumulation of collagen fibers. So we expect there should be increase in anisotropy. That correlated with the findings that we observed here. And also the tissues supported the clinical significance and the chamber specific behavior which were observed in LV and RV fibrotic samples. And here what we also concluded was there were more pronounced changes in the case of LV samples and in terms of the optical parameters, which told us that the LV ventricle fibrosis is a more robust indicator when it comes to looking for rheumatic heart valve disease or myxomatous valve disease. So what were the key points and the research summary which I wanted to put forward is that we were on the page into exploring an underreported pathological condition, which is myocardial fibrosis. Because in general, if we see, there are studies which majorly targets a specific cardiovascular disorder, not a pathological condition. And we investigated the efficacy of developing two optical modality based systems. And based on the research findings, we envisage that whatever testings which we have done on the human heart samples, these can be used as a valuable tools for the cardiothoracic surgeons in the ex vivo scenarios in addition to the gold standard. But the future work always holds the the advanced scope in conducting the study on large cohort of samples and also exploring other cardiovascular disorders, followed by exploring and optimizing the system for in vivo studies, which will always be conditioned to in vivo ethical clearance. But yes, the work that we have done till now, it gives a better insights about an underreported pathological condition, which is myocardial fibrosis. And whatever research we have conducted here, one of the work was presented at IEEE Photonics conference that was held in Florida, followed by conference presentations at Pitch Fest and project exhibit at Kahotech. This work was also presented, few of the research findings were also presented at the Clio Europe conference, which was held in Munich in 2025. And since I told you that whatever study which we have done, it is done on the human samples. So it has to, follow the institutional ethical clearance. So we obtained the clearance from ISC and from the collaborating institute. And these are the clearance certificates. And with this, I would like to end my presentation. So thank you so much. And I'm open to questions.
Thank you. It was a nice talk. Just two questions. First is that you chose the optical mode for investigating whether did you consider any other mode through which this information could have been obtained? That's number one. And this, do you have something like a false alarm rate or detection where the procedure indicates that there is myocardial fibrosis, but in reality it isn't there? Things like that. If you have any information.
So to answer your first question, yes, I didn't just explore optical modality. I also explored electrical modality, which was electrical impedance characterization, followed by characterizing the cardiac model parameters. So in that modality, basically we fabricated a chip and developed a system, but it was for ex vivo characterization, wherein we were trying to see that how the myocardial tissue properties changed at room temperature compared to body temperature. So it was an IDE integrated with the microheater. So we tried measuring the impedance at room temperature, followed by heating the tissue to body temperature and seeing how the properties shift for normal tissue versus fibrotic tissue, and also finding out some cardiac model parameters that related to the tissue heterogeneity. For example, the resistance of extracellular space, intracellular space, capacitance of cell membrane, resistance of cell membrane. So that is the second modality we worked on.
So all of this is done on the tissue. The specific tissue is taken out, and then all the tests are done. So what is the scale factor? Like you will have the complete heart, and you will have to do the test on that, right?
So Twinkle, the question is that on what scale these experiments are done? Like you just take a particular tissue, or you like take the whole organ kind of?
No, she has done on a particular tissue, and the test is done, and then it is done that there is polarization, and so there is some fault with the tissue. So suppose you have the whole heart. So how will you scale that?
So let's say if there is a whole organ like an heart, then how will you scale the findings?
Okay, so basically this is something which is made for an open heart surgery. So it's not like we need a complete organ. The doctor just need to touch that reflectance probe on the heart tissue. So scaling will not be a problem because the entire heart is made of a musculature called as myocardium. So the tissue heterogeneity which we are talking about is inclusive in the entire heart, not just a specific tissue. So because we cannot start directly experimenting on heart, we started with ex vivo characterization, that is excising the tissue out of the heart and experimenting. The next step to this will be, once we do a study on the large number of tissue cohorts, we can move forward to in vivo settings. So that will also give us a better insight about how it is behaving there.
What is the LED she used? What is the safe limit of that LED power?
What is the safe limit of the LED power that you have used?
We have used a laser diode basically. It works at 1550 nanometers. It is a 5 milliwatt power.
And what happens when the heart is beating? The ex vivo tissue is on the table. But in vivo, that's what ex vivo means. In vivo, it's within the body. When the patient is alive and heart is beating, there is a blood flow. In that case, how this optical property will work, how the probe will.
Can we ask now?
Yeah, he knows that question, but yeah.
So did you get that question, Twinkle?
Yeah, I got it. Okay.
Please.
So what I explored was in that case of scenario, because when the heart is beating, there are two ways. One is ventricular expansion, one is ventricular relaxation. So there is something called as ECG gated acquisition. So what we can do is this reflectance probe, it can be mapped to an ECG gated acquisition, wherein when we have the peaks, that is when the ventricles are actually contracting, at that time we are not taking the readings. But when they are in the relaxation mode, then when we get the troughs in the ECG, we can have an acquisition. So then the noise that we get with the beating of the heart, that won't interfere much. But this is something
This
I
seems to
just
be low.
explored. But in actual scenario, we have to test and see how much interference it takes into account or not.